Thank you to the audience for logging in for this Pharma Lab Next event. This is the last panel discussion for today, and we move on then to tomorrow's panel discussion. Dr. VG Naik, who was supposed to moderate this panel discussion, had to be called away on a medical emergency, and which is why I have stepped in to moderate in his place. Moving now to our very, esteemed panelists who have given us time today on this very busy day in the middle of the week. I'd like to move to Dr. Sandhya Shenoy from FDC. Dr. Shenoy, today's panel discussion is on next-gen pharma labs, techniques and technologies. And you've always, been a very passionate advocate of new technologies and new techniques. But I'm sure that there is a lot of, gap between implementing a new technology and actually rolling it out, wanting to implement it and then roll it out. So what has been the experience within FDC as you all moved from a traditional lab into a digital lab? At the workforce level, at the investment level. Dr. Shenoy?
Yeah, a very good evening to everybody, and it's a delight to be here. And normally at all Express Pharma events, and very thanks for the honor. Yeah, as Viveka, as you rightly said, yeah, we need to move forward to new frontiers of enabling our laboratories and our scientists to a greater transparency and better documentation practices. We have been very fortunate that we have seen the last decade has been very good for the pharma industry. There has been a sustained and robust growth and pace. And post-COVID, a lot of interest and of course investment in the drug discovery, vaccines, like medical equipment, biologics, everything. The organizations are looking at ways and means how to maximize profits and also how to reduce the life cycle of a product from lab to the consumer. Now, in this process, as an R&D person, we need to ably support the organization by robust and fast-paced development. Now, current times are very stressful because stakes are very high, competition is very fierce, and so we need to be very agile and very rapid in our development programs. And I don't think the time today, like the need for speed, efficiency, and productivity is today at the greatest height, which was never felt earlier. And COVID has been probably the greatest teacher for us how to change our way of thinking and doing things. Not only R&D, but I think all functions in an organization now need to be more leaner and agile. And since I'm coming from R&D, so what could we have done to improve our efficiency, improve our transparency? And so this was discussed with me and my team, like we sat down for a discussion last year. So how do we improve our process efficiency? And then we decided that probably it's time now that we need to migrate from our traditional lab notebooks, like which writing physical lab notebooks, to maybe e-lab notebooks. So we were deliberating for maybe more than 6 months time, but then we decided, yes, we need to take the plunge. And so we had a lot of discussion rounds with the vendors and all. And last year, not last year, this year, yeah, early this year, we decided we went live with the lab notebook. Now, the reason behind it, we all are aware that we are subject to a lot of scrutiny being in the pharma industry. And probably no sooner than later, even R&D labs would be subject to inspections. So it is better that we have an archival of all our lab work done. So it is one platform which gives you the freedom to the scientists to document whatever work is done, to review the work, and also a permanent archival of the things. So it facilitates easy archival, easy review of the things done. And also you can track and trace what was done, what was changed. So it is good for permanent also in the development process, and good for the organization and good for us also. So we can remove or we can retrieve any information at any point of time. Now understand that since being in the pharma industry, like technology or adapting new technology is a little difficult for us because we work in the regulated space, and anything new needs to be verified, validated, and then evaluated and then implemented. But then this thing was doable at the R&D because it is still at R&D, so it was doable for us, and we decided we need to make this change. So, and it's been more than 6-7 months now. I know the results are not very visible immediately, but definitely all of us are very happy with the things we are doing now and how things have changed from the traditional notebooks to the ELNPs. And I'm sure the benefits probably going down in the years to come, maybe a year or so later. The benefits will be far visible. Like this also allows the scientists some free time, maybe not initially because now we are just getting used to the things, but maybe a few months down the line, the scientists would be more free to engage themselves in more productive work rather than worrying about writing physically in pen and paper. And because it allows him to evaluate entire across spreadsheets, the entire batch data, or compare batches, compare analytical data. And so it gives them some more free time to make informed decisions. So this was how we did a small transition to improve our efficiency towards maybe a more digital laboratory or keep ourselves future ready.
Thank you. Thank you, Dr. Shenoy. That was, a very good review of your process within your organization. And I think what you said about COVID being the best teacher, being a harsh teacher, but yes, a best teacher. Perhaps we look back to this time as a time of great learning and, look back to this time for a lot of learnings in the whole course of our corporate journey. So thank you for those views. Moving now to Dr. Sheetal Kumar Pathak, who's from Glenmark. Uh, Dr. Pathak, if I could ask you, how did your company, Glenmark Pharmaceutical, really have to adapt to rapid changes during pharma testing, which is also part of the topic for today, because everybody knows that Glenmark had to ramp up and launch FabiFlu in a very short period of time. And it was not just in launching a new product and adapting to a new product, but doing it at scale almost overnight, and everything depends on quality in the pharmaceutical industry. So Dr. Pathak, can you tell us about what kind of rapid changes needed to be made to your testing protocols to be able to support this manufacturing ramp up? Dr. Pathak?
Good evening. Good evening, everyone, and good evening to my co-panelist. When somebody comes to know that you are from Denmark, the first thing that comes to their mind is FabiFlu, and that kind of gives us all Glenmarkians a lot of satisfaction, and a kind of sense of achievement that at least we together could achieve something and could actually make a difference to the lives of millions of fellow Indians who were actually struggling to deal with this pandemic that hit us hard last year. So thinking of FabiFlu, one thing that comes to my mind is a quote from Walt Disney, right? Wherein he said, all our dreams can come true if we have the courage to pursue them, right? And so here, like our CMD chairman, Mr. Glenn Saldana, he had this dream of launching this product, bringing this product to Indian market, where we can impact millions of Indians. And we, as a part of Glenmark, our team in R&D, we are also part of this dream, and we could actually achieve it, right? Now, talking about this dream, just having dream doesn't work, right? So you have to be passionate when you are actually trying to work to right the wrong. So here the wrong being the virus that hit us badly, right? So when this project came to us in R&D, I mean that time we were at the peak of pandemic, not peak of the pandemic, I would say, because first wave was still better in a way because there were not many infections to start with, but there was a lot of fear, right? So we were at the peak of fear, and there was a complete lockdown, as all of us know, for first 3 months. So when this project came to us, I just called for a meeting, and obviously it was the first virtual meeting that I had here in Glenmark. So I called for a meeting. We discussed, like how we go about it. That time nothing was there with us, neither the API nor the standard nor impurities, nothing, right? So it was just an idea, and we were in midst of this pandemic. So we are discussing like, how we go about it. The first thing was like, how we go to the lab. Because till then the government authorities had not classified pharma as essential services, right? The essential services was mainly the physicians and medical fraternity and police and all those people. So our admin and HR department, they are working with government authorities to get permission to have some people, classify at least R&D as essential services and have some people in lab. So while they were discussing, we had discussions among ourselves, and I was like, see, this permission will be there, right? In a couple of days, we can have at least few people go to the lab. So I told my team that, like you see physicians, right? They have no choice. They are going there and they are working. So we are also kind of in the same business, right? They are physicians, they treat the patients, we make the medicines. So we don't have choice. We will also have to go to the lab and work, right? So a lot of people agreed. And but what we did initially was, we knew that we cannot send many people, and obviously time was running out. We wanted to kind of launch it yesterday, right? So though nothing was there, but the expectation was that it has to just go quick like that. So then what we did was, we just identified what activities we can actually do from home, right? So all documentation, calculation, all meetings, any advisory meetings, any technical meetings, all those things we just classified that these are the activities that we are just going to handle from home. And we'll make our process, work in such a way that these activities actually can be done from home. And I can tell you that if I consider the whole R&D activities, including the transfer of our technology to our manufacturing location, Baddi, from where initially this product was launched, I think more than 60% of the activities actually we did from home. I myself never went to lab till the launch. I was just working from home. So this is how it all happened, right? And then it kind of as Dr. Shinoy said earlier, it kind of taught us many things, right? This pandemic taught us to think differently. There were a lot of logistic challenges, right? Even to send something from here to Baddi was a challenge, right? So all these challenges kind of taught us to think differently, and that is how many things I think have already changed, and they are going to change gradually in future also. I see that many things in the lab testing are going to change. One of the incident I just want to narrate. We were almost on the verge of approval. We are just about to hear from DCGI, and they actually asked for some additional data. So we again sent few of our people to the lab. They started working. We were running the experiments, and next day we had actually 6 positive cases in the lab itself. So our admin and HR was like, no, the lab shuts down, and 3-4 days no work. We'll sanitize the whole facility. Then only we'll decide on allowing somebody to go. I again called for a meeting of my associates, and we are discussing now how we handle this. So, the first day that we discussed about these physicians, right? So somebody was like, oh, okay, physicians, how they go to the lab, right? They wear all these safety gears, right? So why don't we have the same safety gears, and then we will be allowed to go to the lab? So I spoke to our admin, and they said, yes, if we have those, PPE kits, you can go. So we actually had, they actually arranged 4-5 PPE kits for us. People went to the lab, worked, we submitted the data, and then a couple of days we got this approval. So this is the story about Fabiflu. But now when I see how it impact the lab, right? So if I see any pharma lab today, I think lab essentially consists of 5 different components, right? The most important and the first component is the physical lab itself, right? The lab where you work. The second is the instruments and equipments that you operate, that you keep in the lab to do different type of testing. The third is the procedures or the methods, test methods as we call it in pharma industry. This is kind of an enabler for the lab. Fourth thing is the processes or the systems that govern this lab, right? And the fifth is the human resource who is actually working in the lab. Our lab analysts, supervisors, managers, lab heads, all type of people. So if you really see, all these five have changed in a way and have a lot of scope to change in future, I think. So when we consider the physical lab itself, the first question that comes to my mind is, can it be virtual, right? And when I think can it be virtual, I just look at people from IT or this software industry, right? So when I see them, when I see my neighbors, they are work since last 1.5 years, they are working from home, and next 6 months or so, they are saying that they are not going to go to the offices, right? So I was just thinking, is it possible in our lab? Is it possible in pharma lab? So the first thing that came to my mind was no. But then I saw partially, yes, we can do it partially, at least to start with, right? So as I said, all these documentation, calculation, processing, these activities actually can happen from home also, right? That can be done from home. The next part of the lab is the equipment and instruments. And if we look at any pharma testing lab, I think 70% of the equipment or instruments are HPLCs, UPLCs, GC, LCMS, ICP-MS, all these equipments, right? And all these equipment, all the processing at least, I'm not sure whether in India some labs are doing, but I know that in US many people are doing this. The processing client can be at home, and you can process all the data at home. So once you kind of start your sequences, load the sample, all the other activity you can actually do sitting at your home. The next part is acquisition. That also in few labs actually they are doing it from home. So even acquisition also can be done from home. So 70% of this activity, at least 50%, I think can be done from home. If not now, maybe in coming years we'll see that. So the next part is again instruments. I think Dr. Upadhyay is going to talk more on automation. So automation is already happening, and I think it is going to increase only over a period of time, right? So this automation is going to help us both ways, right? If the things are automated, you don't need to be there all the time physically, right? So that way it will reduce your physical presence. And second thing, again, that is the topic of our discussion today, GLP, right? GLP quality issues. So if you have a lot of automation, obviously all these analyst errors or human error, you will avoid all those. And we all know our whole industry in India is facing a lot of issues. Many industries are facing a lot of issues with FDA and other regulators as well, right? And the most important component of those issues is all these analyst error, human error, because that kind of creates suspicion, right? So we will be able to reduce these human errors once we have more automation, and that way we can reduce the physical presence of people in the lab. Then the third thing that I was discussing was with respect to procedures or the test methods. So procedure or test method, Dr. Sandhya already spoke about digitization, right? Lab notebooks, electronic lab notebooks. So even these test methods and procedures, I think probably 50% of the industry now probably they are using the digitized procedures and methods, right? But there also we can improve, and I think that is something that that will probably coming in next few years. We had one method wherein the procedure is there, elaborate procedure is already given, but still the analyst in QC is not able to execute it properly, right? So what we did in this case is like we added an annexer or addendum to this procedure, wherein we actually added one video, right? The total how to execute the method, the complete video itself. So maybe in future we can have this instead of having these procedures, we can have these videos itself, right? Which can actually demonstrate the procedure. It will be we already are doing this for method transfer, like even from for Fabiflu, as I said, nobody traveled to our plant, right? So it was all video conferencing and showing the demonstrating things from R&D to plant. So similarly, these procedures probably also can go that way as well, right? Right now we have electronic copies, but it can actually go to videography as well. Then the fourth thing is processes and systems. So basically, when we bring about these changes, we'll have to make our processes and systems flexible to accommodate these changes. And last but not the least, in fact, when I was talking about instruments, actually, this is with respect to the automation. But I think our industry is also changing a little bit, right? Earlier we were into generic space. Now from generic to complex generic to differentiated products we are going, right? A lot of companies are working on liposomes, complex injectables, derma. We are already working, but now drug device combination type of products. So here also a lot of characterization is involved, and right now there are certain techniques where we do not have any facilities available in India. If available, they are only in the academia, right? So that is also one of the area wherein, for example, AFM, speed analysis, all those type of techniques probably also going to be part of our labs in coming days, I guess. And then coming to the human resources, I think gone are the days when people were in the lab all the time. I used to work 12 hours, 13 hours, and I was happy doing that. But today's generation, they want to have this work-life balance, and rightly so. So I think if we have this, kind of hybrid, work from home and work at site, offline, and online, partly, I think that will go a long way in, improving our productivity and the human errors that we do, right? If people are happy, obviously, your personal life also impact your professional life, right? So if we want to create positive impact, many companies, I think, Western world, they are already giving these. I mean, even before pandemic, also these options were there, right? So I think now with this pandemic, probably we also learn our lessons and incorporate that hybrid model here also in India. That will go a long way in improving efficiency, improving GMP, and improving quality.
Thank you, Dr. Pathak. That was a very good view of what went behind the scenes at Glenmark for FabiFlu. I'm sure it's very interesting to the people listening in. And moving now to Mr. Balaji from Alembic Pharma. There was a situation that the pharma industry had to deal with a lot of contamination in the sartans. When NDMA was found, and then there were batch recalls, and then very quickly testing procedures needed to be ramped up because the US FDA and all other regulators started mandating different kinds of tests, and basically also said that the amounts that used to be okay to be present, they made them more stringent. So how did Alembic cope with such a diktat? And sartans are kind of a very universal drug, and so I mean that kind of ramp up, not just in manufacturing but also in testing procedures, needed to be done at a very fast pace. How did you do it in Alembic? Can you tell us the inside story?
Yeah, thank you, Lakshmi. First of all, I thanks to Dr. Nayak and the co-panelist to associate me as one of the today topic. Since Alembic is the unmatching a lifespan of 117 years in the pharma manufacturing, and basically I joined on 2009 end, so almost more than a decade I was spent. So we transformed the macrolids to all other generic product, and but when we come to the technology, we need today the timeless management, how to adapt, how to upgrade, not only to upgrade, because technology has come, there are many instruments has come, you can do whatever you want, you can get the quick regulatory filing, but how to understand that, for that you need the skill. Not only you need the skill, but also how to transfer, how to transform to the down level so that it can able to adapt in the manufacturing side also. So this is all the very challenging, and we have to handle with this human resources. So as already the Shetal Kumar Pathak already told that to handle those things, we have to balance their life and we have to make it. So these are very challenging. When we come to this, particularly sartan, Alembic is manufacturing all sartans, right from losartan, irbesartan, candesartan, telmisartan, olmesartan. So all sartans we have been manufactured, we have the ANDA holder. So when we had a discussion, almost I had a discussion with the US FDA and the USP of the USP of the US. where they have published some of the pharma industry, the methodology. But what happened after subsequent the sultans, where they have developed almost 7 to 8 impurities and identified. So this is a big challenging to keep for each impurity for one technique. And also the challenge is different sultans. There are almost 8 sultans. Those sultans, we cannot adapt the company who is manufacturing all the sultans to identify this impurity on a routine quality control. It is a big challengeable. We cannot adapt to the multifold technique because the manufacturing, we are doing so many sultans on the day-to-day for releasing batches. So we cannot do the different technique on day-to-day because number of instrument, number of resources. So even the FDA came out with that, only the limited sultans. So we worked, initially we worked with 2019, we have released the part term, only the selective of 2 or 3 sultan. So then the company requested us how to make it all the sultan single method because we are continuously releasing. So we are discussing with US FDA and USP. Then we worked out called Alembic one method solution. And US FDA came to our office and they sat with us. They discussed almost 4 days. We developed finally after 1 year thorough work, we have developed a single method for all sultan, for all impurities. So this is very challengeable and which we can prove with not only this is by LC-MS technique. No need to change any sample preparation, irrespective of any analyte, irrespective of the any matrix. There is only one method. Preparation is same. Either is irbisartan, losartan, candisartan, telmisartan, or olmesartan. Anything you can do only one sample preparation, and all the 8 analyte can easily be detected to the level of the current guidelines as given as based on the dosage is 0.03 ppm. We are gone up to 0.002 ppm. So then USKP came, USFD and USP together they came. They sat with us of 3 days. They reviewed the record. Then they took the method. They have tested in the US different laboratory. Finally, we had a conclusion. Then they want the full validation procedure. So we have almost in the month of January 2021, we have submitted all the documents. So it's a very challenging. The crux of the main thing is gain the experience of earlier we have developed a GCMS. Then our plant itself having a challenges because GCMS is having its own disadvantages in terms of heating and everything. So then secondly, we cannot able to do it with a single method because of the different sample matrix. So we have constantly worked on the sample extraction procedure and preparation. And then we finally MS parameter. These two we worked almost 6 months. Then finally we come to have a single method, unique method for all the sultans, for all the analyte to the extent of 0.002 ppm. So in that what we gain the experience is, we have to do the skill is, the key parameter here is sample preparation. However, we can able to do multiple way. We cannot do anything on that because the same analyte, same diluent, how to extract the trace level impurity, and that also it should be more sensitive. We cannot add more sample concentration because that will also impact the sample matrix. And retrieving the residual level of 2 ppb. 2 ppb is a very challengeable. So we have done, and how to account for the accuracy in terms of how you get back there. We have done a standalone of each analyte. We have done a standalone. There is a technique where we confirm that what we got it is almost 98% of that. And also additionally, we have done with high resolution, the mass based, with almost 5 ppm Dalton, we can have a degree of accuracy of these analyte. So which confirm that there is no any co-eluting of, because these are all, for example, NDMA is the mass is very low, is almost a 74. If NDA means 1 or 2, so very low mass, there is a possibility of the other the sample diluent mass or some other masses artifact which may. So we are ensuring the 5 ppm Dalton accuracy. We ensure that whatever we are detecting the particular analyte which is having a 5 ppm mass base accuracy. So this challenge, this experience which we have done, what we understand that then the transformation into the plant is another big challenge, because normally in the plant quality control, they are very familiar with HPLC to the extent of PDA and the single, that is a single quad mass based. So a triple quad and MRM mode, everything. Every week we have a training program at our analytical, and we went to that, the site training, and almost we have took the separate one folder and we have trained almost 5-10 people, and then we reciprocate. We made a document, we have submitted to the FDA that now we are capable of that. So those things, now it is become very easy. You can do any certain, any analyte, we can able to achieve. This is the one of the very key challenges which we have faced, and Alembic has got a very good high-end remark from the US FDA, where they are going to implement, because they have got all their scrutinization was going on. Finally, they found that how this method is so robust, and it is a very easy to adapt in the any of the manufacturing plant. That is what our aim. It is not only for Alembic, any manufacturing plant, very easy to adapt to this single based technique of one method solution. So this is one technology. Secondly, the elemental impurities now, we have been transformed from atomic absorption spectrophotometer earlier. Now we have transformed to the ICP OES, called optical emission spectroscopy. Now we have transformed into ICP MS. Now you cannot use AIS, you cannot use OES. You have to do it, the ICP MS. Why? Because for all elemental impurity, drug products and drug substances become mandatory. If you are not using any of the metal intentionally in the process, however, we have to test those elements as per the guidelines of class 1, class 2, class 2A, 2B. Because if some of the excipient or during the process in the manufacturing container or the drug product storage container closure system, there is a possibility of leaching this metal. So this needs to be identified in the ICP MS. Because ICP MS, there is a challenge compared to ICP OES, where we have to do, based on the limit, 30% of that limit, you have to keep it as a LOD. So, which you cannot able to do by ICP OES, because the technique is very less sensitive. So there is no other way we have to do the ICP MS. Again, the challenge is ICP MS sample extraction. It's a very difficult challenge because of the sample matrix of a respective API and the drug product. How to get the trace level that to 30%, for example, arsenic are very, very low. So we have to get 30% piece is only a ppb level. So you have to get and you have to validate. Again, the sample extraction, we have to develop, we have to transform into the plant after validated. So these are all the things where we need to have extensive update on the technique and skill, and people has to work, and then we have to. So this will also have to, people has to, now we have to change our way of working, not only develop, but also developing method as to utilize in the plant utility. So transformation is a very big challenge, the technique and how to make it as a protocol. So this also Alembic is working out. And we have some challenges, but now it is, we need to bond with helping each other in the plant with their mindset. All of you know that how the mindset of the R&D based and plant based. So we need our extraordinary patience, not only our knowledge, but also the transformation. You need a very good alignment and cohesive bonding. That's the most technique. The third one is NMR. Why I'm bringing that, the LCMS and NMR today is not considered to be high-end. It is a quality control technique. We have faced many of the OOS. To identify things, we have to do quickly the NMR. Nowadays, NMR is embedded with artificial intelligence. What we can do it without isolation, the exact sample matrix with the trace level of the impurity with 0.02%, 0.01%, you can able to do LCMS by 2D LCMS and 2D NMR quickly without isolation of impurity. We can able to identify the mass as well as the structure. There are multiple ways in the artificial intelligence called CMC, Constitution of Molecular Confidence. So that program, we can quickly assess the molecular structure and mass. With that, we can able to assess that whether it is related to the impurity with the API, or it's a reaction during the thing, or from the vessel, or it is contaminated from the previous API or drug product. So we have done so we need to integrate the technology what in the so-called R&D. Now it is no so-called, it has to come to the plant. Then only you can prevent many of the OOS to make this center of excellence a key force in the plant, and we can able to integrate very effectively.
Thank you, Mr. Balaji, for that wonderful way that you've showed how your company Alembic has done it in three very important technologies, and how you all have adapted really to evolving regulator concerns when on sartans and other products, and how you all have really kind of done it. And now the next challenge, as you said, is to do what you have done in the analytical laboratory at the manufacturing stage. So all the best to that, and we'll be very closely watching that. Moving now to Dr. Pritesh Upadhyay from Lupin. Thank you, sir. And we've talked about how each company has kind of implemented different technologies, the new generation techniques and technologies. How have you done it at Lupin, and what have been the benefits so far? How much time did it take to kind of get this ROI? And if I could also ask you to comment on the data theft and hacking concern, because we have heard of a lot of pharma companies kind of being hacked for their data, etc., especially the products related to COVID. So how has Lupin really, what kind of firewalls, what kind of precautions have been taken on that front? Dr. Upadhyay?
I think I have 10 minutes time, now almost 5:20.
Yes.
So I won't take more than 10, 12 minutes time.
Absolutely, sir.
So, as a whole industry, if you see, not only pharma industry, is a paradigm shift. The initial people were thought of result oriented. Now it's more towards system oriented. When I say system oriented, there are certain other systems we need to incorporate as a part of the systems. And why system oriented? Because, at the end of the day, you need to see the system should work by itself. If you go by the result-oriented manual systems, you always feel problem and doubt that things works properly or not. And I have seen particularly if the organization is para 4 filings, if you go for any litigation, and during the litigation, you need to submit lot of document to your attorneys. And whenever you collect the document from all the concerned people, if it is a manual operation, that's very problematic. That you are not 100% sure that you have collected 100% document for the relevant product or not. And to eliminate all these things, in Lupin, we decided that what system we incorporate so that first, it should minimize this human error. And we thought that particularly chromatographic, nowadays the software for chromatographic software like Empower software is available who can take care of your chromatographic system or chromelein systems. So all these systems are available for chromatographic system. What about the other system like spectroscopy? And nowadays, because of the people who are working more on complex generic, chromatography is part of the things, and now it's hardly 10 or 20% of the total entire documentation. If you see 70 to 80% documents are from different techniques. And hence we have decided that why not to think to synchronize this laboratory data. And for that, we decided to implement LIMS. And before starting LIMS, actually this activity was started somewhere around 2018, 2019, and decided that which limb system is appropriate or suitable for the Lupin. And for that, we have done various proof of concept, and then we finalize one of the system which we feel that it's more useful and helpful as an organization. And let me tell you that what are the benefits so far, because now this limb system we have already implemented and it's working almost 80 to 90% completed, and people are using day in, day out. Some of the things which I particularly experienced, that the biggest advantages is of elimination of transcription error through this instrument integration. Another thing is that supporting this is the compliance regulatory FDA 21 CFR 11 compliance. Another biggest thing is I found is the single electronic repository system for all the operational data. Otherwise, what happened, earlier we need to collect all the chromatographic file after file, and there was no space available at our premises. We need to take the help from some other writers, suppliers, so that they can store our documents. So after implementation this, I'm not saying that it is 100% paperless, but very less paper now in laboratory. If you see, this entire system is automated audit trail. So you need not to worry about that what the person has done in the second shift or night shift. Apart from all these things, we also realize the advantages this, tracking of inventory of laboratory material. And even in many cases, it automatically trigger and inform the purchase department that this inventory is going to problematic within a week's time or 4 days, so that they can manage and arrange the stocks. Another thing is that managing test results, flagging out of the specific result quickly and identify abnormal observed values, because all the information available automatically and tracking by themselves. Trending, even stability data also, it can trend and it can predict the tentative self-life based on the available data. The biggest thing which we feel is that secure and restricted access to the data, leading to for better data privacy and protection. Monitoring entire laboratory environment, and even if you see the calibration of instrumentation and maintenance, it will automatically trigger at the time it's due. Another thing is that it is very difficult for a person like me, if you have more than 300 instruments running at a time, that out of 300, which are the typical problematic instrument and what was the issue or whatever the problem you observe. So while incorporating or by implementing these LIMS, it will definitely provide all the trending data and information that which specific instrument is giving more problem, what type of maintenance people have done, service engineer. And even you can discuss with the instrument manufacturer also, that out of your 20, 30 or 100 instruments, these are the typical instruments we use enormous problem, so that at least we can identify the root cause and take corrective action. Another thing is that faster, precise and accurate delivery of result. Nowadays, if you see in synthetic chemist, the people, once they submit the sample for reaction monitoring, they need to know that what is the conversion of step 1 to step 2. So earlier, once you complete the reaction monitoring, then you submitted data. Now they can see online rather than getting any information from the chemist. And they also can come to know that, okay, what is the sample has submitted on yesterday, how many tests so far completed, which are in process or progress, and when I'm going to get the result. So rather than interacting with the intervention of any human errors, the people can see directly. And in a way, if it is all this automation, which definitely reduce OSOT. And that way I can say it will reduce the cost and indirectly you are increasing the productivity. So these are the information which I feel are most advantageous so far after implementing this link system. The another question which you asked about the firewall, to be very frank, so far I'm not 100% sure that whatever the corrective action we have taken, even then we are prevented from any such malware attack. So that's all from my side. If you have any further question, any point, I'll be ready to answer.
Thank you, Dr. Upadhyay. Moving quickly back around the panel, I mean, I was attending the other sessions during the day, and a lot of questions have come on the data security point of view. So, do any of the other panelists want to comment on this point? Dr. Pathak or Dr. Shinoy or or Mr. Balaji before we end this session?
Though,
You're mainly talking about.
One point I would like to highlight that, the first thing people have to do, although I think all the organizations, they might have done it, that when the instrument you are connected with any software, your laptop or computer, that should be dedicated one. There should not be any Google or any other server or any other explorer available on the day, so that people won't search for anything outside except laboratory software. There should not be anything available.
That's a very good point. Yes, Mr. Pathak, Dr. Pathak, you were also, yes.
Yeah, I agree with what Dr. Upadhyay said. So basically, in most of the companies I worked earlier also. So what we do is, like, the acquisition server and the instruments that are attached to this server, right? Say for example, Empower server. So the computers that we are using there, those computers, it will be good if we keep them free of any other softwares, right? Normal internet, or even for that matter, Word or Excel or any other thing. So those stations should be solely dedicated only for running the instrument. That is one of the thing. And other thing is, like these days anyways, I think most of the GMP labs, right, at least in our manufacturing locations, what we are, because by regulation itself, we have control access, right? Only with username and password, only each individual, those have access to those instruments only can actually access those instruments, right? So most of the instruments that earlier were not on server and were not controlled by user access privileges, those are also these days are all coming on that security. So that way it gives us a kind of security, right? And then for all these instruments, I think PLCA has kind of become it has become kind of compulsory. And then you have administrator, software administrator, and usually it is a IT guy, like who knows everything about it, who actually can only access these instruments, with full access. I mean, otherwise it is like controlled access to the users depending on what their role is. So that way, kind of part of security, I think part of it is secured, right? So now all other things, I think, yeah, probably IT and these people are best people to comment on how to save from these practices of stealing data and all those kinds of things.
Right. Dr. Sandhya, if you would like to comment on that.
Yeah, not regarding data security, probably, whatever I think Dr. Upadhyay and Dr. Pathak has said, I think organizations are definitely doing their bit to prevent data breach. I would like to touch upon another aspect, and more so because probably the inspiration came from one of the Express events I attended. So one more area of technology or updation which we are also exploring here is using simulations. Yeah. so far, probably simulations, whatever we have been hearing from childhood is probably what, if simulation comes to my mind, it's rather maybe the crash test for cars or maybe tire burst test. But yeah, we were probably never aware that we could work upon in understanding more better about our processes or making them more robust or preventing failures when we maybe transfer them from one scale to the other. And this happened because one of the products we faced a slight issue when we transferred it, rather not transferred, it was still in the lab stage only. It was still in development. But when we scaled up, like it did not behave the way which we thought it would go, because it was perfectly all right in the laboratory. And in the meantime, I happened to attend that event, I think, which you were, quite this conducted. And so we are early in our discussions with one of the experts, and we are exploring this technology. And I think many organizations are doing it, but still it is still in the nascent stage where pharma is concerned. But I still think there is a lot of potential probably, and it would save at least formulation developers a lot of time and energies, which is invested when we transfer our process to the plant scale. And from this could be one area where we can work more better to save time and I think, yes, even resources.
Yes, so it will definitely shorten the time to market. Simulations will definitely, kind of shorten your time to market. Thanks for making that
Yes.
point, Dr. Shinoy. Mr. Balaji, before we end the session, any parting comments from you on? I know that you spoke quite extensively. So any parting, comments on literally a lot of the panelists have said that a lot of the learnings that they have taken from COVID are going to be implemented, and in your case it's implemented across industry because the US FDA has kind of given a very positive comment on your technique developed. So any parting comments before we close the session?
No, but everything's fine. Nothing to
add to that. Thank you. With that, I'll close the session, and I'd like to thank our panelists, Dr. Pritesh Upadhyay, Dr. Sanjay Shinoy, Dr. Pathak, and Mr. Balaji for their wonderful experiences that they have shared about turning challenges into opportunities. And as Dr. Shinoy said in the beginning, COVID has been a very tough teacher. But I think India Pharma Inc. has responded and been kind of like the ideal student, turning all these challenges into good lessons, into good learnings. And I'm sure that in the days to come, we will see the industry building on these strengths, not just to improve productivity, not just to improve compliance, but the overall benefit of this will be to improve and build on the reputation of the sector as a guaranteed producer of good quality, affordable medicines and therapeutics for the world, not just for India. On that note, thank you so much. And I would like to really thank Dr. V.G. Naik for putting together this panel and for putting together all our thoughts on this panel. I just stepped in because he had to head out for a medical emergency. So thanks to Dr. V.G. Naik also, who kind of guided this panel to this conclusion. Thank you so much.